no objection. I'm starting record. Okay, so I just want to first of all start by acknowledging the folks who have done the work before we begin. Can I ask the folks from my lab who are on to turn on their videos? So basically, Shushan has been working on the single cell ATAC data. Kai has been, and maybe wait doesn't say your name. So Kai has been working on the deconvolution data, and then Ben has been working on the linking. They'll be here to correct me every time I make a mistake and jump in. I'll just very briefly give an overview of sort of the types of activities that we've been working on. And basically, what we're trying to understand is how genetic variation leads to phenotypic variation in order to ultimately enable therapeutics. We're starting with genetic variants, and the goal is to sort of uncover target genes and new therapeutic avenues, and personalized and precision medicine. The challenge, of course, as you all know, is that the vast majority of these disease variants are non-coding, which basically means we don't know the target gene, the causal variant, the cell type of action, the relevant pathways, or the mechanisms. So what we do is that we augment these disease data from common and rare variants with RNA variation across individuals and epigenomic variation across individuals, both in healthy and disease samples, looking at DNA accessibility primarily at the single cell level, epigenomic modifications at the bulk level, and of course, RNA. Then we integrate the data to predict driver genes, regions, and cell types. We're building a lot of deep learning models, Bayesian models, motif-based, mediation analysis, etc. And then we're predicting targets, and we have a large number of collaborations where we're testing these targets in cell cultures and also in animal models. And then, of course, we disseminate the results. So the goal of circuitry is manipulation, of course, as we've previously shown, by uncovering the circuitry underlying disease loci, such as we showed here in the FTO obesity locus. We can now start altering individual nucleotides, or we can start overexpressing or downregulating genes with dramatic consequences, being able to switch between disease and healthy phenotypes. This has all been at the bulk level. What I'd like to tell you about today is what we're doing at the single cell level. So we've been doing a lot of single cell RNA-seq. We basically profiled something like 1,500 brains post-mortem, something like 10 million cells across brain, but also probably another million cells across many other disorders. And we're integrating the results and disseminating, and you've seen some of these papers out already. So our first paper was in Nature last year in collaboration with Ligue Tai. The folks leading this were Hans Reddy Mattis and Jose Davila Vilderrain from my lab and Liwe's lab. And what we basically found was 84,000 cells across the brains of 48 individuals, focusing on prefrontal cortex, looking at both non-Alzheimer's individuals who have very low pathology, shown here in blue, and pretty fine cognition, shown here in red. And then you can see as they get older and more sick, you basically have this progression towards less cognition and lots of pathology. What we found is that the primary drivers of variation were primarily dissecting cell types from each other. And then if you went to the cell types and then you subclustered those cell types within each of these major classes, what you found is that the subclasses of excitatory, inhibitory neuron, astrocytes, oligodendrocytes, OPCs, and microglia were in fact very strongly associated with disease subclusters and non-disease subclusters. So basically low amyloid, high amyloid, early Bragg stages, late Bragg stages, low cognitive decline, high cognitive decline, and then high AD, low AD. And that was quite remarkable. It basically says that genetic variation, even though it might act initially in a small number of cell types, as was previously shown for microglia in Alzheimer's, the impact on gene expression is actually global and seen globally in each of the major cell types. The second lesson was that female individuals have many more sick cells than male individuals, suggesting that they have a higher level of transcriptional level pathology. We've expanded this work to look at somatic variation directly from the single cell RNA-seq data using the SMART-seq technology that does whole transcript. And we're able to see accumulation of somatic burden in specific genes and specific pathways for individual cell types, and specifically excitatory neurons and oligodendrocytes, where we have the most cells and the most power. We're able to actually see the somatic mutation clustering in specific biological processes. Here's an example of schizophrenia. We just posted this paper on medRxiv just last week, and I can put a link. But what that allows us to do is look at a large diversity of cell types, not just excitatory versus inhibitory, but distinguish different classes of layers of excitatory neurons, different classes of inhibitory neurons, including some low abundance classes, and study the gene expression changes associated with each class of neurons. And this is, again, prefrontal cortex, about 800,000 cells. We found more than 20 different cell states. I want to highlight one cross-cutting cell state, which was very cool, this SZTR state, which we call for transcriptional resilience, which I'll explain why in a second, is in fact cutting across layers 2, 3, but also layers 4, 5 of excitatory neurons. So this is a very unusual cell state in that it cuts across multiple cell types. Why are we so excited about this state? Because if you look at the individuals who have this state, and I've shown them here in arrows, they are actually some of the most transcriptionally normal individuals. Let me explain. We basically build a predictor of transcriptional pathology from the gene expression patterns of every individual. We basically ask, how are the expression patterns of that individual correlated with the expression changes that we see in the whole group as we shift between schizophrenia and control? And the schizophrenia individuals are on the right side of that predictor, and the control individuals are on the left side. There are some exceptions. This control individual shows pathology that is very much like schizophrenia, but it turns out that his son is actually diagnosed with schizophrenia, and it's possible that the father escaped diagnosis for some other reason, but his expression patterns are in fact quite predictive of schizophrenia. But on the other side of the spectrum, we had some very, very cool findings. So these SZ individuals are all showing an abundance of this SZTR cell type, which appears to be shifting the expression patterns of all of their other cells to a more transcriptionally normal state. And that's why we call this SZTR state a transcriptional resilience state. And it seems to be highly correlated with transcriptional resilience in these PV cells that are in fact showing these brain rhythm kind of patterns that might be contributing to the normal expression patterns of all those other cells. Anyway, it's a hypothesis, but very compelling and something that we are pursuing experimentally.
So, Manolis, can I ask a question?
Of course.
It's all very cool. Yeah, it's all very cool, but it's a very basic question, which is, how come you didn't actually get all of these subtype of excitatory neurons and so on in the previous analysis you were showing?
So it's a fantastic question. So basically, over here, we're using V2 technology, and over here, we're using V3 technology. Over here, we were using very early versions of our method. Over here, we're using ActionNet. So ActionNet is a new method we just published, again, I think last week, or maybe 2 weeks ago, in Nature Communications or something. Mohammadi et al. And I think it's the combination of, number one, we got better at doing the experiments. So Kiki Galani and Julio Mantero and a lot of other people in my lab and also Liwei's lab have just done thousands of these experiments. So we're frankly better at it. Number two, these samples were younger samples. So basically this is schizophrenia. The other one was Alzheimer's. We're looking at a 100-year-old brain that has been frozen for many years versus a younger brain that has been frozen for less time. And number three, the new technology, V3 chemistry versus V2 chemistry from from 10x. And we're also doing, I think, slightly deeper sequencing and better algorithms. So, and also we know how to use these algorithms better, and there's many iterations of cleaning up that goes into doing that. So fantastic question, Ali, but the answer is like the whole kitchen sink.
No, I understand that. But yeah, I mean, because I would predict that the previous one should have looked more like this.
Yeah, and it probably does with our new methods, but actually, it actually does with our new methods. Okay. Yeah, so part of it is methods.
Okay, that's great.
Manolis, this is Mark. I have a question. So obviously this is very good, and I'm very interested in this. I'm looking at the bottom left-hand corner figure when there. So basically what you're saying is that, just to sort of put it out there, you're sort of saying that you basically get some sort of false positives, but you think they make sense because of this SCTR type of thing. Is that what you're saying?
That's exactly right. Yes.
Okay.
Yeah.
Yeah.
You can call them false positives, of course. Basically, this is a false positive and these are false negatives. Basically, we're predicting normal, but these guys are actually schizophrenic. And here we're predicting schizophrenic, but these guys are actually normal. Turns out his son is schizophrenic. And here turns out that they have this SCTR cell type.
And you said this is polygenic transcriptome score. So that this is polygenic, like a polygenic risk score from their genetics and also a transcriptome score combined?
No. This is polytranscriptomic score. So it's
polytranscript, just transcriptome, no polygenic.
No genetics.
Okay.
Yeah, the genetic story is not as clean as I would like, and in general, the PRS scores that we're using are not, correlating with either diagnosis or transcription very well. And it just might be that we're still underpowered for schizophrenia to use PRS, very effectively at this stage.
Okay.
Yep.
Manolis, you're calling this resilience, which to me implies causality. Do you know, is the resilience signature causing you to be healthy even though you have risk? Or is it rather the consequence of not having disease? Like, for instance, have all these people had a psychotic episode? Or is this the ones that have had versus the ones who have not, or something like that?
Mike, we've struggled with this enormously. And I've constructed models in my head for exactly the opposite, for how the transcriptionally normal individuals would eventually, as a consequence of being transcriptionally normal, lead to the cell state. And every single time I've tried to do that, I've arrived at an inconsistency. So I have not yet arrived at an inconsistency in the other way, where it is actually causal. But it could just be correlation. But I can't picture a way in my head that this would be reverse causation. But it's a fantastic question, and I want to spend hours discussing this over beers, each in our own beer garden. But yeah, I mean, we don't know if it's causal or not, but that's what the name indeed implies. The name implies causality, but we don't make claims of causality. We basically, it could just be that it's a biomarker of transcriptional resilience, that somehow these guys are transcriptional resilient for other reasons, and this cell state is a biomarker of that resilience.
Hi, Manolis, this is Rag here. A simple question. How exactly did you call this X SZTR state? Because it seems part of a bigger cluster, and none of the other clusters have been dissected into subclusters. So what was the process?
So unfortunately, Shakeen, who did this analysis, is not here. But my understanding is that the clustering is orthogonal to the topological placement of the cells. The topological placement of the cells is basically coming from this action net algorithm, and the net part is a network of the global relationships of those cells to each other. As we're moving from one cell to the other, we're traversing a graph of all of these cells. And the ambiguous cells are the ones that you'd see here in sort of lower like shades that are not as strongly shown. These are the cells where the graph is a little ambiguous. In this particular case, the graph for those cells was not ambiguous, but when you do an unsupervised clustering of those cells, it actually picks up two additional cell states. One is this NRGN cell state, and the other one is this neurogranin basically cell state, and the other one is this SZTR state, the transcriptional resilience state. So and I've probed Shakeen many times about this, and I don't think I have a clean answer for why was it picked up as a separate cluster when in fact in the two-dimensional embedding projection of those cells and in the graph, these cells don't lump together as a densely connected module. So I don't know the answer to that, but it's a great question. Okay. So the next thing that we can do is basically now start looking at what are the GWAS loci for schizophrenia. So this is a Manhattan plot turned sideways. I guess this is a drunk Manhattan plot where all the skyscrapers are moving to the right. But every single one of these loci has now been annotated for whether there are differentially expressed genes between schizophrenia and non-schizophrenia individuals in that locus. And we found an enormous enrichment for GWAS loci. But again, that doesn't mean that the differentially expressed gene is the target in each case. We could be wrong in some of them. But what we've reported is, number one, what is the gene that shows the strongest differential expression nearby for each of those loci? And we capture about 50% of the schizophrenia loci. Number two, what is the directionality of effect? Red means associated, higher expression is risk-inducing, and blue means higher expression is protective. And then what is the cell type where that expression difference is seen? And we also show the expression difference in blue or in red, positively associated or negatively associated with schizophrenia in each of the columns for each of the cell types. And you can see that this SZTR cell type is in fact, quite prominent. You basically have multiple genes that are sort of differentially expressed in SZTR, but also primarily excitatory neurons are differentially expressed, and to a lower degree also inhibitory neurons. And most of the time you see these genes differentially expressed in a subset of the cell types, not all of them. And most of them are not captured at the bulk level. And some of them are in fact, showing opposite effects, where it is differentially expressed in one direction, one cell type, but in the other direction, another cell type. And I don't think I have any examples here, but across a larger set of differentially expressed genes, even outside the GWAS associated loci, you basically see such evidence. And then here we have high-C evidence for linking between these loci and the gene that shows the differential expression. So you can see that for about 50% of them, we also have high-C evidence. For the rest of them, it's just co-localization of these. Happy to take any questions.
But Manolis, how do you know that the differentially, you're saying that the cell types have genes that are differentially expressed. How can you differentiate that from just more or less of those particular types of cells in those individuals?
Oh, because this is single cell RNA-seq data.
But you know that those cells that have been clustered are lower in expression, but they're still clustered to that same cell type.
No, gosh, of course, yes. So basically, the beauty of single cell data is that it's not deconvolution. Basically, with deconvolution, we would have no idea. Basically, if we were just deconvolving bulk data, we would say, yeah, it could be abundance, it could be cell type abundance, or it could be cell type specific changes in expression. But now we super clearly have the cell types, and then we can say, okay, for this cell type, how is the expression changing for that one gene?
Okay.
Okay, so then we can do all kinds of cool stuff. We can basically say, for all of the genes that are globally differentially expressed in schizophrenia, can we find out what are their predicted master regulators? So we basically look at the enhancers of those genes, we look at the upstream motifs, we look at the regulators corresponding to those motifs, and we can actually start asking, number one, what are the differentially expressed genes themselves, the targets, enriched in? Number two, what are the regulators enriched in? And what we're finding is that the targets are enriched in synaptic signaling, synaptic organization, and neurodevelopment. And you can see here how this enrichment breaks down across the different cell types for both upregulated genes and downregulated genes. You can also do that for the master regulators. You can basically say, what are the enrichments for those? And what we see is that the enrichment of the master regulators is a little more specific than the genes themselves. So the regulators appear to be acting a little more in neurodevelopment and to some degree synaptic organization, but not to the same degree as the genes themselves. And then we can actually start clustering these regulators into modules based on their transcription factor expression correlation across different neuronal cell types, and then sort of construct these modules here. And then what we're finding is that there's this very cool module, which was also picked up by scENCODE as module 37. And this module basically has 4 regulators that are both schizophrenia GWAS associated and also lead neurodevelopment transcription factors. So basically these are the purple ones. And then these guys are enriched in neurodevelopment, but also in synaptic plasticity. And these guys are sort of enriched in these genes that are shared both in embryonic and in adult tissues from scENCODE. So we can get some hint as to where do they act along the developmental time course. So basically this is kind of cool because it basically finds these set of regulators that are controlling both the synaptic component and the developmental component, which are generally, a little separate. We've done this kind of analysis for many traits. I'm not going to mention all of them, but basically for ALS and frontotemporal dementia, we basically looked at the motor cortex, and we're now doing the prefrontal cortex to basically look at sort of an affected and an unaffected tissue for each of those. And we're finding a lot of pathways that are enriched differentially between ALS and FTD, and what target genes are associated with those, what pathways are enriched, and what are the cell types where they're acting. We've done this in Huntington's disease. This paper was just published a few months ago, looking at this direct pathway versus indirect pathway spiny projection neurons, which are quite distinct in the cellular identity prior to Huntington's. But with Huntington's, you basically see the loss of identity. And indeed, this is one of the cell types that is thought to be strongly associated with Huntington's. We've also started looking across different brain regions, looking at the midtemporal cortex, entorhinal cortex, angular gyrus, prefrontal cortex, or anterior thalamus, and hippocampus, across both AD and non-AD individuals, and across both RNA and ATAC, so a large number of samples. And we've basically constructed these sort of mega, ActionNet plots across 1.6 million cells in this particular case, looking at all of the different cell types and how they're changing across brain regions. And what we're finding is very surprising. We were expecting that neurons would be very region specific, but we're finding that nearly every cell type except for microglia appears to have a lot of region specific expression patterns. So if you look at even astrocytes, something that we would not expect to be very dramatically different, you see that the subcortical versus the cortical regions and specifically thalamus and mammillary body are in fact showing dramatically different expression patterns for those. And we're also seeing that these tissue-specific expression changes are in fact correlated with tissue-specific pathology. So we can actually go and measure pathology in each of these regions of the brain in the ROS-MAP cohort in collaboration with David Bennett. And we're finding that the region-specific pathology is in fact correlating with the expression changes in those regions. Very excitedly, we're finding that there's a lot of spatial information that is captured using from those single-cell RNA-seq profiles. So if you look at this picture, you basically see that the excitatory neurons are in fact subclustering into many different subclusters. And those subclusters are in fact capturing a lot of the spatial organization of the hippocampal structure, including the subiculum, CA1, CA2, 3, 4, and the dentate gyrus, which basically indicates that single-cell data, in fact, is capturing some aspect of the spatial data. And that's very exciting because what we are indeed seeing is that there's quite substantial differences in the correlation with Alzheimer's disease between these different areas. So CA1 is in fact the most predictive of disease, and dentate gyrus is the least predictive of disease, suggesting that we can now not only find, in fact, the pathways associated with AD in each of these sub-regions is actually quite dramatically different. So basically what we're starting to do now is building spatial catalogs of marker genes that we can then start using, or I shouldn't say marker genes because these are global expression patterns, but basically spatial catalogs of global expression patterns that we can use to now, spatialify or something, basically sort of augment single cell data with spatial data. And we've basically built some deep learning models that allow you to do that, that basically allow you to sort of position our single cell data using spatial transcriptomic references. And this is, ongoing work. We've also started looking at coordinated changes between single cell RNA and single cell ATAC in Alzheimer's disease. So here's some examples where you can see, for example, different genes in different cell types that are coordinately changing in expression and in accessibility. Here's ApoE, for example. You see that it goes up in disease and in expression, and it also goes up in accessibility between control and normal individuals, and you can, non-AD individuals. And then you can see the same thing for astrocytes and this gene here going down in both, and so on and so forth. And then you can see the coordinated changes between single-cell RNA-seq and single-cell ATAC-seq across multiple genes in this combined plot, where the arrow basically tells you how both RNA expression and DNA accessibility change coordinatedly in AD versus non-AD. We're also able to look at cell type specific enrichments for our single cell ATAC data. And this is work by Shushen, who's also on the call. And basically what we're seeing is that if you look at the GWAS enrichment across all of the single cell ATAC peaks for each of the cell types, you see that Alzheimer's enriches very nicely in microglia, and only in microglia. This is something that we and many others have published before multiple times, but it's the first time that we see it using single cell ATAC data. We had previously seen it with CD14 plus monocytes from ENCODE, but now we're actually seeing it with, and we had also seen it in cell sorted data, but now we're seeing it in single cell data. And then for psychiatric disorders, you see the enrichment is primarily in excitatory and inhibitory neurons. Again, as you would expect. If you look at random immune disorders like ulcerative colitis, cirrhosis, etc., you see this enrichment in microglia consistent with the immune nature of microglia. Not suggesting in any way that they're implicated in these disorders, but you also see that for non-brain disorders, you don't see these enrichments as a control. And then what's really cool is that you can actually go into those locations and then find these microglia specific peaks and then look for motifs that are repeatedly disrupted in these regions by the SNPs that are localizing in microglia, but also in each of the other cell types. And what we can do is actually start predicting potential regulators whose motifs are repeatedly disrupted in the single cell ATAC data from each of the cell types. And here's one example here of SPI1, which is this immune microglial master regulator that whose motif appears to be repeatedly disrupted, and that is itself an AD-GWAS target. We're also able to
enough power for this, Manolis?
Sorry, say it again.
You're getting enough power to assign that with confidence?
To assign the regulators, yes, because we have multiple loci where they are repeatedly disrupted.
Okay.
Yeah. So the power comes from the many loci. For the single cell ATAC differential accessibility between AD and non-AD, we are suffering from power. So basically, there's only a few hundred loci that we can find as significantly different between AD and non-AD. And I expect that there are many more.
P.1 has been published as a GWAS hit for Alzheimer's disease.
Yes, absolutely. So basically, this is something that we reported in our Joneska paper in Nature in 2015. We basically had said, conserved epigenomic signals in mice and humans reveal immune bases of Alzheimer's. And in the abstract, I think we had also mentioned P.1, which is, again, a well-known regulator for...
Well, I mean, P.1 and SPI1 are the same thing. That's
yeah. I'm sorry.
That's why I'm bringing it up here, but yeah.
Yeah. So basically, SPI1/P.1 is both a master regulator and a direct hit, which is kind of cool. So basically, and it also is a hit of itself. So it sort of contains P.1 motifs, suggesting some kind of loop as well. So thanks for pointing it out, Ali.
Yeah, and it's the key TF for microglia, right? For macrophage identity and stuff like that. This is
a marker of differentiation. It's actually what we're using when we did the sorting experiments for K27 acetyl. We basically sorted using P.1/SPI1. So given our interest in microglia and given this an enormous enrichment for AD, but also given that one of the top hits for schizophrenia, as beautifully shown by Beth Stevens' lab and also Steve McCarroll's lab, is in fact a complement gene suggesting some kind of microglial role in synaptic pruning associated with schizophrenia. We also said, can we somehow use our data to actually subclassify microglia into subtypes of microglia? And one of the things that we're finding is this synaptic microglia versus these inflammatory microglia. And what's really cool is that the inflammatory microglia are enriched for GWAS variants associated with Alzheimer's, whereas the synaptic microglia are associated with GWAS variants associated with schizophrenia. And if you look at the differentially expressed genes using scRNA, you basically find that the inflammatory microglia, so differentially expressed genes in Alzheimer's between AD and non-AD individuals, whereas the synaptic microglia are showing differentially expressed genes in schizophrenia, which is something very cool. So basically, it seems that we can now take this microglial association and localize it specifically for inflammatory functions in AD, but in fact, synaptic functions in schizophrenia. So we've done also a lot of work in single cell ATAC and GWAS integration. So basically, taking all the single cell ATAC data and then calling cell types from this, classifying the cell types, and then looking at the cell fraction and how that changes by AD, sex, etc. And then looking for genes that are differentially accessible in each of the cell types, and then looking at the peaks that are differential between different cell types, and then assigning these cell type specific peaks in each of those, and then looking at their corresponding enrichment. And then, here's some beautiful examples of how cell type specific many of these peaks are. And now we can start asking, are these cell type specific peaks enriched in these specific transcription factors? And again, we find this PU.1 and SPI1 figure that I showed you earlier. And we can also find hundreds of differentially accessible peaks from single nucleus ATAC-seq for each of the cell types. So basically, in excitatory neurons, you basically find this enormous number of differential peaks because that's where we have the most power. And in microglia, as you mentioned earlier, Mike, we are a little underpowered, but we are still finding these differential accessible sites. And we're also finding these different functional enrichments for these differentially active, differentially accessible peaks for single cell ATAC, where we're finding enrichments for neuronal and secretory pathways in excitatory neurons, enrichments for a lot of immune factors and immune signaling in microglia, and then for general brain functions in astrocytes and so on and so forth. And then corresponding examples for the fine mapping for each of those. And then I showed some of these results earlier.
Manolis, this is Mark. So just explain. So the stuff that you're talking about here, do you from the single cell ATAC data, do you call the peaks? You have a peak caller and so forth, from the single cell ATAC data.
Shushen, could you answer this question, please?
Yeah, we basically assemble all the reads from each cell type and do the peak calling. Yeah.
No, that's fine. The reason I'm asking is because there's been a lot of discussion. I just wanted to highlight to you, but also to other people on, presenting, because you obviously didn't present the details of how you do that, but people are very interested in, comparing that to Snap ATAC and, Archer and all these other things. And so that might be a good thing to do another time. I just wanted to put it out there.
Perfect. Let's add that as an agenda item. Sorry, Shushan, go ahead.
Yeah, so this analysis was performed using Archer, and I also tried a Snap ATAC. Both of these tools are performing great, but yeah, this was performed using Archer. Yeah.
Okay, great. Thanks, Mark. Thanks, Shushan. And then we also looked at QTL analysis to basically find genetic variants that are associated with differential accessibility. And you can see here some examples in each of the cell types where for excitatory neurons, for example, or inhibitory neurons, you see this differential accessibility based on the genetic variation at these loci. So again, this is the kind of thing that can allow us to now get at causality rather than just correlation associated with the disease. And you can see here the localization specifically near the ATAC sites, which gains confidence for this. And again, with microglia, I was expecting to be underpowered, but it seems that we are able to find many of these loci quite well. So the last thing that I want to touch upon is deconvolution, and this is work that Kai Kang is doing in our lab. So Kai developed this awesome method prior to joining our lab called CD-seq, which is basically taking bulk RNA and then without any reference, this is not reference-based deconvolution, this is without any reference, looking at the matrix of expression by sample, and then using that matrix to separate out both cell type proportions in each sample, as well as cell type specific expression profiles cutting across these samples. So again, if the data is clean enough, you should be able to decompose it into a small number of archetypes and then their proportions and how they're changing. So that's what scATAC does. Again, this is something that he published prior to joining our lab. And it's a Bayesian network model that is basically inferring both these proportions and the cell type specific expression patterns de novo based on that variation. So he's able to demonstrate using these two examples that it actually does work, and it's able to capture these specific archetypes of these cells and actually separate out those cell types across using both synthetic data and simulation data. And he's now started using that. And I think that's something that's very relevant to ENCODE and specifically to NTEX, where we've basically now started deconvolving the GTEC samples using this approach and looking for cell type specific EQTLs across the GTEX cohort. And we'd love to apply that, of course, in NTEX. So this is looking at the frontal cortex from GTEX, and every dot here is the single cell expression pattern of the deconvolved single cell profiles that Kai is discovering in the bulk data in conjunction with all of the single cell RNA-seq data. So we're basically plotting jointly the major cell types and the deconvolved cell types. And what you can see is that not only is he capturing each of the cell types, moreover, he's capturing the diversity of expression in each of the cell types. So the thicker points are places where he's discovered that cell type de novo without any kind of reference from the data. And the reason why we're doing frontal cortex is because that's the place where we have an enormous amount of single cell RNA-seq data, so we can systematically validate this. We're also doing this in heart, and we are also doing this in several other cell types where we have ongoing collaborations and ongoing single cell data, and it seems to be performing extremely well. These are the cell type proportion estimates from the frontal cortex samples from GTEC. So he's basically deconvolved all of these samples and finding these proportions, which again make a lot of sense, biologically based on what we're finding. And he's doing that across all of the different regions of the GTEC's brain data. And he's, again, discovering these proportions. And you can see here, for example, in the spinal cord, you see a lot more of these endocytes, whereas in different regions, you see just these changing proportions exactly as you would expect biologically. And now he can start looking at how many E genes do we discover, how many genes that have EQTLs with the original data versus with the deconvolved data. And it's quite remarkable. So he's able to, dramatically increase the number of E genes. And again, the beauty of all this is that genetic data is completely orthogonal. If his deconvolution was bogus, we would not be finding genetic signals that correlate with the cell type specific data. He's also been using his approach for spatial transcriptomics. So we've started using the Visium spatial transcriptomics platform in our lab to basically start looking at the spatial context of all of these. And what we're doing now is treating each of these little pucks, each of these little spots, as a mini bulk experiment. And this is a beautiful setup because scATAC, the deconvolution method, is actually exploiting the fact that you have slightly different combinations of the same type of expression patterns across many, many different replicates. And that's what every one of those spots gives you. It basically gives you a slightly different composition of astrocytes and microglia and oligodendrocytes and so on and so forth in each one of these spots, enabling us to now start learning these cell type proportions. So we can basically, for example, using the spatial information, start looking at the proportion of excitatory neurons in different spots of these 2D slides placed on the spatial transcriptomics map, and looking at the astrocyte proportion. And then looking at the oligodendrocyte proportion. And this is very cool. It's something that in single cell data, you just just dissociate all these cells, and then you have individual cells. You would never be able to see that, in fact, astrocytes are sitting in the middle, surrounded by excitatory neurons, surrounded by oligodendrocytes. And here you can see the expression of the corresponding genes, marking these territories very nicely. You can also do that using layer information and here spatial data where we're actually deconvolving the expression of each of these marker genes of different layers. And indeed, you can see these deconvolved cell types placed along these layers. Here's a projection of the deconvolved cell types for each of those. And you can see how it very nicely agrees with these layer 2, 3, 4, 5, and 6, which are peaking at different spots along this gradient of expression. We've also started looking at the spatial changes that are happening in the context of disease. So basically using these spatial transcriptomics of these SCA1 mice, which are known to be associated with changes in these Purkinje cells. We're basically looking at the spatial transcriptomics here of the wild type mouse and the SCA1 mouse. This is data that Kiki Galani generated just 2 weeks ago in our lab. And you can see now these, spatial information and how it's associated with the structure of the underlying data. And he's now able to deconvolve each of those little locations and discover this, basically the deconvolved results are capturing the diversity of Purkinje cells. So here we're showing both the spatial transcriptomics data that we generated and deconvolved in the bold cells, and also the single cell data that Kiki also generated for the same matched sample of both wild type and SCA1 mice. And you can see that we're capturing both the diversity of the cells and the abundance of the cells. And what's really very cool is that if you look at the PPP1R17 gene, which is a marker for these Purkinje cells, you see no expression difference of that marker between wild type and the SCA1 disease model. However, and also in the spatial organization, you see these same sort of bulges in the mouse cerebellum in both a wild type and the SCA1 model. However, the moment you deconvolve rather than just look at the expression of the marker gene, you see this dramatic difference between the proportion of deconvolved Purkinje cells in the SCA1 mouse versus the wild type mouse. You see this dramatic reduction, which is simply not visible if you use marker genes. So deconvolution is really adding information that is simply not visible if you just try to do marker gene-based expression, and also the spatial organization seems to be quite dramatically altered for those deconvolved cells. The last topic that I wanted to touch upon is how to link.
Actually, Manolis, just before you go on to, I know you're running out of time. I just wanted to quickly say we're certainly happy to have a discussion on the NTEX with the single cell QTLs and the single cell deconvolution, with that CD-seq or whatever. I think that'd be fun. We'll follow up with you with the.
We'd be thrilled to contribute.
But the one question I have, and also I know this is a little bit of a loaded question, but I'll just ask is, if the if the reference list deconvolution works so well, why do you need to do single cell data? I mean, can you do everything with the reference list deconvolution, or is there certain things it doesn't do?
I will answer in the exact same way I answer every time somebody asks these types of questions. I mean, we're showing deconvolved data is doing great. We're showing that imputed data is doing great. And people are like, oh, great, you can just impute all of ENCODE. Why don't we just stop here? And I'm like, no, that's ridiculous. So I know that you're asking this rhetorically, and I'm going to answer, as bluntly as I always answer, which is absolutely, there's so much stuff that you will never be able to capture using the deconvolved data. If you look at how much are we capturing of the diversity, it's a tiny fraction of the diversity. There's all this stuff that we would be missing if all we did is deconvolve. So, I mean, of course, I'm not advocating for let's stop doing this, but what I'm saying is that we're going to basically, and sort of this is just demonstrating the power of de novo deconvolution, but reference-based deconvolution is potentially even more powerful. So the reason why we absolutely need to continue doing these experiments is because we will get better references. We will, of course, get better validation for these methods. And we will also able to capture the diversity of cell states much more cleanly than deconvolution will ever get at. I mean, yes, for some applications, when samples are missing, and because these samples are just so much more expensive, when you're using spatial transcriptomics, you don't have the luxury of doing single cell. When you're using, I mean, there's just so many settings where you can't do single cell because of technology. Eventually, you will be able to do it. But I see the two research programs as moving in parallel. You have to do a ton more single cell because you're learning so much more than you will ever get by deconvolution. But you also need to always deconvolve because you get so much more information that you don't get from the mini bulk spatial transcriptomics period. So you need both. And the other thing that I very briefly mentioned earlier in the talk is that we are not only able to deconvolve the small number of spatial experiments that we're doing, but using these experiments, we can now start spatially placing a ton of additional single cell data that we are generating. So you could say, oh, great, if you can infer the spatial from the single cell, why bother with spatial? Again, you need both. So I'm certainly a computational person, but I have enormous respect for how much better every single one of these methods gets with more data. So we are nowhere near a data saturation point. We are at the infancy of data generation. So we need a ton more data. I hope that's the answer you were hoping for.
It's very good, Mel. Let's just keep going because I know we're running out of time. Very good answer.
Okay, great. So I couldn't be more blunt about that. So, nowhere do I mean to supersede data generation. All right. So last topic, just very briefly. So linking GWAS variants to their target genes. I mentioned briefly that we've generated this EpiMap resource that's combining roadmap and GGR and ENCODE. We now have 800 reference epigenomes. For each of those, we have chromatin states. For each of those, we have enhancer calls that combine the inaccessibility enhancers and the peak in K27 acetylation signal. Using these, we can now take 2.1 million enhancer elements and then condense them into 300 patterns of expression, patterns of activation across the different tissues. So we now have these 300 modules, and they're enriched in specific processes, which actually makes a lot of sense. If you look at the gene ontology terms of nearby genes, you basically see a lot of enrichment for each of the tissues for those processes. If you look now upstream at who are the regulators that control them, you can use regulatory motifs to basically predict who are the upstream regulators for each of those modules. And again, the results make a lot of sense. And you can actually start looking at, focusing on specific biological tissues like heart and immune and brain, and look at this enormous diversity and high resolution inferences that you're making for each of those tissues. We're able to intersect all of that with genome-wide association studies to basically find enrichments in 200 plus traits using just raw epigenome enrichment, and 540 traits using looking at these tree-based enrichment. So if you look at the location in the tree, we're building a tree of how are all the samples related to each other, and then using that tree to look at enrichment in different nodes of the tree at specific resolutions. And that actually is a 10-fold increase from the 54 GWAS tissue enrichment that we had found in roadmap. So huge increase in the number of tissues, huge increase in the number of GWAS enriched traits. We are able to look at the the network of trait by tissue association. So placing traits in a network according to the tissues in which they are enriched and co-enriched. You can see here these psychiatric and IQ associated traits localizing in brain. You can see Alzheimer's and a bunch of immune traits localizing in blood. You can see heart and blood pressure, etc. But you can also see CAD here, one of the most diverse traits, which is associated in this pie chart with tons of tissues. And we can actually now use EpiMap to start decoupling all of these complexity by partitioning the subset of loci associated with CAD that are overlapping liver enhancers versus coronary artery enhancers. And these are showing different gene ontology enrichment, and these are showing different comorbidities with other tissues. we're also using all of these to start linking enhancers to their target genes. And Ben James, who's on the call, is responsible for this work in collaboration with Carlos, who led the EpiMap study. And we're able to ask how many genes are associated with each enhancer, and there's about 1 to 5 genes per enhancer. How many enhancers per gene? There's about 30 enhancers per gene. How many links are in each tissue? And you basically have 3 different groups as the median and up to 7 groups as the mean for the number of tissues in which every link, every enhancer gene link is active in. We're finding that links are even more specific than enhancers. They're about 80% more specific than enhancers. So enhancers are very specific, but links are even more specific. And then the enhancer gene average distance is about 90 kb. And longer links tend to be more cell type specific. So if you look at, the 2 sides, they're quite different. We're able to evaluate these links using many different methods, such as gene ontology enrichment with the nearest gene or without the nearest gene. And our methods are dramatically outperforming other methods using this approach. And we're also outperforming or performing comparatively to other methods using multiple different types of validation approaches. So all of that is in the revised EpiMap paper. So how are we doing all this? So again, this is work that Ben developed in collaboration with Carlos. He's basically looking at correlation between the chromHMM enhancers and their activity, and looking at the correlation with nearby expression measured as FPKM or log FPKM. And then when they correlate, you basically use this correlation as evidence across multiple tissues, and then comparing that correlation distribution to the distribution that you would expect using random genes. And you're also taking distance into account. So, we're also using both the correlation as well as the activity of these elements to get at the cell type specificity of the links. And then we're using distance as a model parameter because distance is in fact very highly correlated with every other prediction method. So I mentioned some of the performance, and the other very cool thing that I think will be very generally useful is that we have these gene ontology based validation of gene enhancer links using Go enrichment to gauge the performance. Why is that useful? You can basically take a set of enhancers that are linked to those genes and ask, are these enhancers enriching in, you can use grades to look at what are the gene ontology terms that the enhancers are enriched in, take those gene ontology terms, and then look for genes that belong in these gene ontology terms as positives for your linking. Namely, your algorithm is not using the gene ontology information at all when taking an enhancer and linking it to a nearby gene. But what you can do is ask, as I link this enhancer to this gene, is this gene belonging in the gene ontology class that this group of enhancers belong in? And the group of enhancers is enhancers in that tissue or in that module. So that allows us to now have these gene ontology-based validation, which is quite orthogonal to Hi-C, QTLs, any kind of proximity, and so on and so forth. So, using this method, we can in fact validate these quite nicely. And again, we're finding that the enrichments for those are dramatically higher, and we can use this type of validation for GWAS loci as well. And we've now made all of these links available for 30,000 GWAS loci in this EpiMap website. So for every one of these loci, you can basically, for every one of these studies, you can
share maps together, exploring enchanted forest.
For every one of these traits, we can look at all of the GWAS loci that are associated with the trait. And then for the top, second, third, fourth most enriched tissue, we can ask, are there enhancers in that locus that overlap that tissue? And what is the distance, which is the number here, between them? And for every one of those, you can click and get the specific association between the GWAS variants and the links to the nearby genes and the tissues where those links are acting. And you can see just very cool examples. Now I'm going back to the CAD study that I showed earlier, which enriches both in liver and in heart. And you can see the associations for some liver only, heart only, or both liver and heart loci. And all of that is available through the EpiMap website, where you can find here compile.mit.edu/EpiMap. It's all on preprint in bioRxiv, and hopefully will be appearing soon. And oh, we're also doing a lot of high throughput validation. I'm not going to go into that. So that's our slide for today. I'm happy to answer any questions. Again, this is immense numbers of collaborations with many super duper labs, and the folks doing the work are the folks here. It's just that none of them is willing to speak as fast as I am willing to speak. But if you have any questions, we are super collaborative and happy to oblige. So any question for the linking part or for any other part?
I had a question about the enhancer gene linking part. That was really great. We've been working on collating more CRISPR data from our own experiments and from other ENCODE groups, and would love to compare to these types of links. I'll follow up afterward.
We are enormous fans, and I'm so excited to hear from you, and we would be delighted to participate. So yeah, let's do that. Sounds great. Thanks, Jesse. I was hoping you would say something.
Well, we are a bit in overtime right now.
Okay.
So but it sounds like your slides are going to be available and your papers are out there. So wow, a lot of stuff here to think about, Manolis.
Thank
thought
you.
myself. Great.
And I pasted the link to the slides at the beginning. For those of you who missed it, I'm pasting it again. Here's the links to both the PDF version and the PPTX version of the slides. Again, big kudos to Ben, Kai, and Shushen and Carlos. Carlos just showed up. Carlos, do you want to turn on your camera and say hi? They are the superstars doing all this work. I'm just a guy who's organizing their Zoom calls. And then Jackie is also here from our lab. She's doing awesome work in cancer, in metabolism, and in Down syndrome. And Nai is also here, big superstar from our group. She's working on all the microglial work that I mentioned briefly. And then Alec Chapman is also here, and he's the one doing all of the convergence work that I unfortunately didn't have time to talk about, but we're happy to come back again at some future time.
Great job, Manolis. So I think we, any other final questions? I think we should end it because we're at our past time.
Okay, awesome. Thank you so much, guys. Please email us questions, and for any point of interaction, we're happy to collaborate and go back and forth. I also see Li Lun here, who's on the experimental side of our lab, coordinating a lot of the data generation.
Right.
Okay. Thanks, everyone. Bye-bye.
Bye.
Bye-bye.